Hello friend, today we are going to talk about gel electrophoresis, the concept of gel electrophoresis, the process and the requirement of gel electrophoresis. Agarose gel electrophoresis is widely used technique for the analysis of nucleic acid and protein. Agarose gel electrophoresis is routinely used for the preparation and analysis of DNA. Gel electrophoresis is a procedure that separates within an electric current. DNA is negatively charged. When placed in an electrical field, DNA will migrate towards the positive pole anode. An agarose gel is used to slow the movement of DNA and separate the movement of DNA and the separate by size. If we talk about how fast will the DNA migrate depend on the size. Small DNA move faster than larger DNA. What is agarose? Agarose is a linear polymer extract from seaweed. Agarose was first used in biology when Robert Koch used it as a culture medium for tuberculosis bacteria in 1822. Making an agarose gel. An agarose gel is prepared by combining of agarose powder and buffer. These are the two buffers, tris acetate EDTA and tris borate EDTA. Well, tris acetate is used for longer DNA like 1500 base pair, and tris borate EDTA is used for lesser than 1500 base pair of DNA. Okay, these are the requirements for electrophoresis. Equipment, power supply. We need power supply. We need gel tank. We need casting tray, and we need electrical leads and gel combs. Preparing the casting tray. Seal the edges of the casting tray and put into the comb. Place the casting tray on the level surface. None of the gel comb should be touch the surface of the casting tray. Agarose gel preparation. Combine the agarose powder and the buffer solution. Agarose is insoluble at room temperature, as you can see in the left side of the picture. The agarose solution is boiled until clear, as you can see in the right picture. Gently mix the solution periodically when heating to allow all the grains of agarose to dissolve. Be careful when boiling. The agarose solution may become superheated and may boil violently. It has been heated and too long in a microwave oven. Pouring the gel. Allow the agarose solution to cool slightly, approximately -60°, and then carefully pour the melted agarose solution into the casting tray. Avoid air bubbles. Make sure that single bubble will not appear. It may cause the traveling. It may cause affect the traveling of DNA sample. When the cold, the agarose polymer is forming a flexible gel, as you can see in a light blue color. It should be appear light in color when completely cool, and it will take approx 30 to 40 minutes. Carefully remove the comb and the tape. Place the gel in the electrophoresis chamber and enough electrophoresis buffer to cover the gel to depth at least 1 millimeter. Make sure each well is filled with buffer. Sample preparation. Mix the sample of DNA with sample loading buffer. These allow the sample to be seen when loading onto the gel and increase the density of the sample, causing them to sink into the gel well. Loading buffer. There are two types of loading buffer: bromophenol blue for color and glycerol, which is used for the weight. Loading the gel. Carefully place the pipette tip over a valve and gently expel the sample. The sample should sink into the valve. Be careful not to puncture the gel with the pipette tip. Place the cover on the electrophoresis chamber, connecting the electrical leads. Connect the electrical leads to the power supply. Be sure the leads are attached correctly. DNA migrate towards the anode when the power is turned on. Bubbles should be formed on the electrodes in the electrophoresis chamber. After the current is applied, make sure the gel is running in the correct direction. Propofenol blue will run in the same direction as the DNA sample. Okay, staining the gel. For the staining the gel, we use mostly people use ethidium bromides. Ethidium bromides bind to the DNA and fluorescence under UV light along the visualization of DNA on a gel. But ethidium bromide is powerful mutagenic and it is moderately toxic. Gloves should be worn at all the time. Safer alternatives to ethidium bromide. You can use these ethidium bromide because they are safer and inexpensive, less toxic, no UV light required, and no hazardous waste disposable. Disadvantage: less sensitive, more DNA needed on the gel. When you are doing your experiment, DNA is very precious, and the amount of DNA or cDNA is very less. So most of the researchers don't want to lose their sample for these for any kind of experiment. So it's very precious. So this is the best. This is the worst disadvantage, that more DNA needed on the gel. Longer staining and de-staining time it will take so much time. Dye requires an ethidium bromide requires an ultraviolet light source to visualize. As you can see this in the picture. So this is the gel after the running of the sample, and here in this gel we use ethidium bromide. You can see this the wells, and the ladder. Some people say ladder, and in the right and left side some people say ladder, and some people say marker. And here I label with the according to base pair, 100 to 5000 base pair. And as you can see that I mentioned positive, negative, negative, positive. So you can see in the sample where sample in here only 1 2 3 4 samples are present. Visualizing that this is another one picture of the gel. Thank you. For more video I would like you please subscribe this channel, and after this presentation I will give the introduction trailer research associate YouTube channel. Thank you so much.